Thank you, Sandra. Yes, welcome everyone. And as Sandra presented, indeed, we are talking today about p-values, and we heard already the first questions and first reactions about what are p's. And as Sandra mentioned, I'm leading the statistics team in the Department of Clinical Research. So to start again, do you know P? Another title to my talk could be a call to embrace uncertainty, and we'll see in a moment what is going on. So we are part of the Department of Clinical Research, and I think in the hospital, most of us who are doing science, or most of us who are, or part of you who are medical doctors, surely aim to give the best evidence-based medicine. This is the key word in our medical practice today: the conscientious, explicit, and judicious use of current-based evidence in making decisions about the care of individual patients. And we know that evidence-based medicine is based on, well, this evidence in evidence-based medicine is based on clinical expertise, patient values, and the best external evidence. In other words, what we generally want to do is okay, I see I may need to speak a bit louder. What we want to do is to base our decisions on data, or on science. And in this case, and we could say that science in this case is what we can find most likely in books, in scientific papers and journals, etc. And then the question may be, yes, how do we get data or evidence published so that indeed people can get it? When I started my studies in my bachelor's, the dean told us in the very first chemistry lesson, if you do science and you don't publish, it's as if you haven't done anything, which is to a degree correct. You need people to know what is going on. But now I'm going to provoke your thought and ask you, what is happening? Could it be that most published research is actually wrong? And I'd like to, this is of course a provocative question, but I'd like to raise it and show you why I'm claiming that this potentially might even be true. So let's look and see what is happening. When I start doing some research, let's assume that we have 1,000 possible hypotheses, possible associations that I may be able to discover or not, or to investigate. This could be associations between risk factors and a disease. This could be effects of a biomarker or effect of a treatment. And there could be, of course, many more than 1,000. But let's, for sake of example, assume that there are 1,000 such hypotheses that I might be able or be interested to examine and test. Now, note that now in this example, I'm going to assume that we're doing everything sense. So statistically, we're going to be using the correct methods. We're going to have research that is correctly or powerful enough to detect effects when they exist. And we're going to use a significant level or a type 1 error level as commonly used. Everything will be done correctly. So in this sense, let's see what happens. We have 1,000 hypotheses, potentially. And let's assume that 10% of these hypotheses or of these associations that I'm investigating are indeed true. So these are the green hypotheses here on the bottom. Out of the 1,000, about 100, for our example, are true hypotheses, and all the rest are negative associations or non-existing hypotheses, incorrect hypotheses. So I said we're doing everything right, and I said we have a power to detect things. Typically in clinical research or other research, often we take a power of 80%. This basically means we have an 80% probability to identify from these true hypotheses the correct one. So 80% of the true hypotheses I indeed detected with my significance test or with my hypothesis test or with my statistical method that I used correctly. So these I managed to publish, and here are the 80 out of the 100 that I indeed published. Note this immediately means that 20% of these have not been identified. So these are true hypotheses that haven't been identified. We could call them false negatives. We did not identify them, but this was wrong. We made an error here. What about the rest? Well, typically, we take a significance level or a type 1 error level rate of 5%. So out of these negative results that we had, assuming we could do these 1,000 trials or studies indeed, then 45 of these would give me a signal even though they're not correct. I would get a signal there, the 5%. So these are the false positives that I may get. So 45 plus 80. Now, what about publishing negative results? We often talk about that. Well, we know that depending on the field, between 10 to 30% of all negative results might actually get published, but of course that's not a large amount. Let's say we have here some more true published negative results. So where does this bring us? It brings us actually that all we see in actually published research are only this part of all the potential hypotheses that existed out there, which I don't know how many there were, nor do I know how many were indeed correct. But if we reorganize this, we can easily see that what is actually published consists of about 2/3 that is correctly published as we identified, either negative or positive results, but about 1/3 is actually wrong. And of course, we do not know which third this is. We do not know which of the studies is really true positive and which is a false result for us. So already, now this is based on the fact that we used what I said, correct statistics, and used the common correct. We did it right. We took a type 1 error rate and we investigated this using a p-value as we wanted. Looking at this in a different way, I mean, what we actually want to do is identify these correct hypotheses. Now, I would argue that logically, if a hypothesis is correct, I expect that once I do it, the same study again, I should get the same result. So a group of scientists from psychology, basically this open science project, they took 100 studies. This was one of the first investigations of this effect of replication. Took 100 published studies from psychological research and tried to replicate them. And indeed, from the 100 studies, 97% of them showed a significant result in the original published manuscript. When this group of scientists replicated the study as best as they could, based on the study protocol and everything that they could find from the manuscripts, well, they only succeeded to replicate and get a significant result again on only 35 of these studies. So this is what started leading to what people maybe heard of the reproducibility crisis. We cannot reproduce a lot of the science that we did based on and using these kind of methods, saying, let's look if something is again significant as it was in the first paper. So what is going on? Is all our science wrong? Are our methods completely off? Are we misleading ourselves completely? Well, I'd like to offer today that one of the problems, or at least one of the culprits to our confusion and to the fact that we are not so sure how to go on here, is the p-value. And we're going to see why, and this is the topic of today, actually, of course. But to understand why I'm arguing this and how this is related, we should maybe look, first of all, at how do we get research published. Let's start with what most of us have learned. Also, I learned this when I started my scientific career or my studies in science, is, well, when I have a hypothesis, I come up with some hypothesis. I say, I believe that the world is like this. So this is my research hypothesis, to which I define a null hypothesis, saying, well, there's no difference. There's no effect to what I believe is going on. I go out, collect data, and then I run a statistical test. And then for my statistical test, I examine whether the P is lower than any conventional threshold that I define for myself. Commonly taken, P 0.05, at least in clinical studies, medical research, or ecology, or psychology, and everything. And then, I mean, the whole process, how did I define my research process? What is my null hypothesis? What is my alternative hypothesis, if I even have one? The way I collected the data, it all boils down to this question. Was the p-value below 0.05 or not? This is my ritual. I either go out to the bar and celebrate with my colleagues that I managed to publish and I found my research hypothesis to be correct because I could reject some null hypothesis. Or, as Sandra also presented in the beginning, I go and cry in the room and, well, go and try again with a new hypothesis, maybe if I'm persistent enough. And then I build myself this arcane p-value to aspire to, and I pray that the next time I do a research, my p will be below 0.05. Indeed, not joking, several authors already call this null hypothesis significance testing the null ritual. And this indeed turned into some ritual. I develop a hypothesis, I collect the data, well, I define some kind of null hypothesis, collect data, and pray that it works, and otherwise my scientific career is almost gone. And note that we lost all this information here. What is the research hypothesis? What are the alternatives? How was the data collected? What was the statistical significance in reality, and not only this yes or no p-value? And we know, of course, as we said before, what is published and what is not published. Actually, let's look at that. What is published? Well, here's a citation I found not so long ago in July from Twitter, where Professor Nicola Raihani cited some decisions, some comments from an editor on a paper she tried to publish with some other co-authors. First comment, both reviewers as well as myself felt the main weakness of the manuscript is the fact that it reports negative results. So it's not worthwhile to publish this because the results are negative. This is kind of counterintuitive. If it's negative results, it's also important. Another comment to the same paper, given the lack of significant findings in the experiment, meaning probably p-value was not smaller than 0.05, and the discrepancy with past search or research, I would suggest sending this paper to a more specialized journal. In other words, we found something that doesn't fit with our current view of the world, and let's not publish it. And this is probably based, again, due to some p-value that was probably larger than 0.05. So this is about what gets actually published. What kind of hypothesis and what do we understand? Well, here's the jelly bean problem. Here, scientists, someone claims that jelly beans may cause acne. And comes the research company or the producers of jelly beans and say, well, let's research this. Well, we can check if yellow jelly beans cause acne, or pink jelly beans cause acne, or maybe blue. And every time we'll just check whether p-value is smaller than 0.05. And no, not with green, not with magenta. Not with blue, not with magenta, but oh, the green ones, we got a p-value smaller than 0.05. Let's go to the press. News article, green jelly beans linked to acne. You may have heard of p-hacking or these kind of words. This is indeed exactly an example of that. And unfortunately, we know this happens. Moving on, what about how do we analyze data? Well, this research team led by Brian Nosek, they actually said, let's check that. Let's get one data set and examine, and this data set simply takes and examines the question whether or not football referees give more red cards to players of darker skin colors. And they sent it out to over 29 research teams and allowed over 61 people, experienced analysts, to analyze the data as they see fit and report to their findings. And these are basically the results of these different analysis methods and analysis teams. And you can see quite a bit of variation in the estimate, quite a bit of variation in the confidence intervals or whatever measurement of variance to the estimate that is given. 9 teams found no significant result with the level of 0.05. 20 teams found a result. What do we understand from this? Can I trust this result or not? Is this a true effect or not? And indeed, I mean, just looking at this, 20 out of the 29 did find that referees tended to give more red cards to dark-skinned colored players. But can we trust this? What is correct? What is the truth? We don't know, just based on this. But I do think a nice thing about this is that we see the direction, at least we see when it is more or less repetitive. And we can draw some kind of conclusion, but we do see that there's variability here. And it also depends on the analyst team and analytical methods. What about how we interpret our results? Here's a published paper from BMJ, parachute used to prevent death and major trauma when jumping from aircraft, a randomized controlled trial. Indeed, they approached, screened 92 people and got about 23 people to agree to be randomized to jumping from an airplane with or without a parachute. And here's the p-value I find for death or major traumatic injury. P-value of larger than 0.9, we found no effect. There's no problem jumping from an airplane, parachute or no parachute. Well, obviously, it's not only the p-value. I think it's obvious that we should also look at the context of this study. I'll tell you now that this is from BMJ Christmas edition, which give us some interesting and funny aspects of science. But the point really is that many times we look at the p-value, decide for a conclusion, disregarding the background and the context of the study. Again, it all boils down to just the p-value and whether or not we published. So we can even come back to our example from before. I mean, in the end, the question I want to ask myself, even if I now publish my study, and I showed you before that potentially, even if I did everything right, about a third of what is published is wrong. And what I, as a researcher, want to do probably is to make sure I'm not making a fool of myself, that when I publish something and say, hey, I found an effect, it is indeed true. And as we said, we typically take a p-value that's going to be small or equal, maximum equal to 0.05. But the truth is what, as I said, as we saw before, some of these p-values will be wrong. Some of these effects will be actually false effects. In other words, what I actually want to know is what is the probability that the results I'm reporting are false positives? Or in other words, will I make a fool of myself if I publish something and then in the end it's actually wrong? What we are actually interested in a way is how many of the studies that I'm publishing are false from all these all these green signals we got here. This is, if we count from the example we looked at before, this would be the 45 that were wrongly identified as an effect, divided by all those reported with an effect. The 45 plus the 80 from before, which brings us to 36%. So even by reporting my p-value, my study with a p-value of below 0.05, I have a 36% of reporting something wrong. This is what we call the false discovery rate. I have quite a high percentage probability to report something wrong here. Some of you may realize that this is somewhat similar to diagnostics and screening tests where we have sensitivity, specificity, and what we actually are very often interested is a positive predictive value. Indeed, the false discovery rate is the complementary of the positive predictive value. And this is sharing the same concepts from there. So, as I said, a lot of what I showed you here boils down to the question of what am I doing with this p-value? And indeed, this is a major culprit. we raise the question, should we stop using it at all, completely? Well, the whole discussion and also the examples I gave you and this whole thought context led in 2016, the American statistician or the American Statistical Association to come out with a very important and large statement, the ASA statement on p-values in terms of context, process, and purpose. Just some quotes from this statement. The statistical community is deeply concerned about issues of reproducibility and replicability of scientific conclusions. We observe that much confusion and even doubt about the validity of science is arising. This is obviously a problem if we want to provide evidence-based medicine or simply to advance in our knowledge. They also identify that misunderstanding or misuse of the p-value is only one cause of the reproducibility crisis, but it is a very important one. It's a very major cause of this. Another quote, the scientific method of testing hypothesis, the null hypothesis ritual from before, is based on very flimsy foundation. And there are numerous deep flaws in this method of null hypothesis testing, significance testing. And our aim today, and what we hope that we all come out today with, is the three maybe take-home messages in a way, that you will have proper understanding of the p-value, that you will understand that proper understanding of the p-value is complex. P-values are not simple to understand. It's important that transparent, honest, and complete reporting of studies, context, alternative hypothesis, all of this is essential for us to draw correct conclusions. And maybe most important for us, embrace uncertainty. Don't be despaired if the p-value is smaller than 0.05. Understand that science is hard and we need a lot of information in order to draw conclusions that will be valid. That is the conclusion of my part. Soon after Sandra will talk a little bit, we will have the two more parts to this talk where Andrea Vincic will talk about, I pronounced it wrong, I think. What is the p-value, and also very importantly, what it is not. And the final session would be from Fabrice Helfenstein, who will talk about what not to do or what to do with respect to p-values, some recommendations from our side. With that, I'd like to thank you for your attention. And I give the word to Sandra while all of you can maybe enjoy some interesting citations of what happens when p-value is not quite below 0.05. Sandra, the word is yours.